Hello, General Biology. This is the second video of 2 for our Unit 3 material. The first video covered cellular respiration and catabolic reactions. This one is going to cover anabolic reactions, specifically photosynthesis as our example. So, as you can see on our screen, we have an overall reaction of photosynthesis. So just as a review, cellular respiration is C6H12O6, which is glucose, plus oxygen, which yields water, carbon dioxide, and energy. So basically, the energy that's contained within the bonds of the glucose over here, as they are broken apart, the energy in those bonds, remember energy is not created or destroyed, it's just changing its form, is now captured in the form of the ATP. So when we look at photosynthesis, we can actually see that it is the exact opposite of our cellular respiration. So we have carbon dioxide plus water is yielding glucose, water in this case, and oxygen. So let's talk about what's happening in both steps, and I'll make sure to bring out where photosynthesis and cellular respiration have a similar mechanism. Okay, so just as a review, this is the electromagnetic spectrum, and you can see that we have broken out the visible light here, but it's a very small slice of the electromagnetic spectrum. We have gamma rays on one end and radio waves on the other, and just to point out to you that visible light is one of the few wavelengths that we can tolerate a great deal of. Gamma rays, X-rays, even UV, microwaves, those kind of rays can be very damaging to cells. Clearly, visible light can also be, but we do seem to tolerate that better than we do the other wavelengths on the electromagnetic spectrum. As you can see, we have visible light, and all visible light in those wavelengths together when combined are white, but when we break them apart, we can see that we go all the way from red here at the longest wavelengths, all the way down to violet, which is at the shortest wavelengths. Notice that these are in nanometers. We will come back and use this information in a couple of weeks in lab. We'll do a photosynthesis lab, and we will be using a spectrophotometer. You'll use spectrophotometers throughout your time in college doing specific experiments, but just keep in mind this information will be useful later on as well. Okay, so this is not physics nor is it chemistry, but I do need to remind you about one of the very specific and interesting characteristics of light. Light behaves sometimes as a particle and sometimes as a wave. For our purposes, we're going to be speaking of light as a particle, and the particles of light are photons. So just remember, when we speak about a photon, we're talking about a particle of light. Now, photons can do three things. They can be reflected, they can be transmitted, or they can be absorbed. Now, when a photon is reflected or when it's transmitted, meaning it passes through, it doesn't have any potential to do work. However, when it's absorbed, it can do work. The photon actually disappears, and it transfers the energy of the photon to the molecule that it has been absorbed by. Now, this only happens to molecules called pigments. And when we think of pigments, I know you probably are thinking of colors. which is true. Pigments do have a color. But let's talk about why they have a color. Pigments do not absorb all of the wavelengths of the light. So when the sun is shining and the photons of light come in and strike the chlorophyll, which we see here, that white light that is being absorbed, or the white light that is striking the chlorophyll, only a small portion of the electromagnetic spectrum is actually absorbed. And as you can see here, green light is actually reflected. Green light is about 600 nanometers. Red light, yellow light, in that wavelength, those are absorbed. So you see green because those are the wavelengths of light that are being reflected. So chlorophyll appears green because it reflects green light, but it absorbs blue and red wavelengths. So when we view chlorophyll, the pigment chlorophyll, it appears green. That means that when you see anyone in a SWU blue shirt, that means that the blue wavelength of light is being reflected and the other wavelengths of light are actually being absorbed by that shirt. So here, our spectrophotometers in the lab will actually do this kind of thing, and we're looking here at the wavelengths that are being absorbed by the different molecules. So you can see that chlorophyll A and B have two absorption peaks here and here. So one is in the red area of the red area of the spectrum. The other is in the blue area of the spectrum, and there is little to no absorption in the green area of the spectrum. Again, that's why it appears green. So that is telling us where those pigments, what wavelengths will allow the pigments to do work. Remember, if a photon is reflected, it has no work capability. Only photons that are absorbed are able to do work. And so that's why we care about this absorption spectrum, seeing that in the red and the blue areas of the spectrum is where those photons can do work. Okay, so here's a picture of chlorophyll A. Chlorophyll B varies slightly in the sense that it has, in place of the methyl group, the CH3, it has a CHO in this position. Chlorophyll B is an accessory pigment. Chlorophyll A, when we talk about chlorophyll, that is the main pigment that we're speaking of. But I want you to see that they have similar structures. Now, first of all, notice that we have lots of carbon and hydrogen and oxygen here, but we also have a magnesium here in the center. That's unusual because usually our organic molecules are composed of carbon, hydrogen, and oxygen. But magnesium is important in this porphyrin ring that is present here. Now, this is where the light or the photon is actually being absorbed and able to cause a cascade, which we call photosynthesis, which is where work is happening. Now, as you can see, we have this long hydrocarbon tail over here. It might remind you of a fatty acid somewhat. This hydrocarbon tail is actually anchoring this pigment into the thylakoid membrane because they're not floating around, they are in a fixed position, and the hydrocarbon tail is what attaches it into the membrane. Okay, we also call these groups photosystems. The book likens them to an antenna system. One photosystem is composed of chlorophyll plus the accessory pigments, so that could be chlorophyll A, chlorophyll B. It could also be other pigments that are not appearing to us as green, which is why the leaves appear yellow and red in the fall, because the chlorophyll degrades first. So this reaction center with chlorophyll is surrounded by these accessory pigments. And so a photon of light will come in and hit the accessory pigment, and when it does that, when the energy from the photon is absorbed by the photosystem, we have excited an electron. And that excited electron bounces around until it eventually goes to the chlorophyll A molecule, where an electron is sent up to the reaction center. Now we're going to talk more in just a minute and look at some diagrams about where that excited electron is going and what it's doing. But remember that the energy from the photon, when it's absorbed, is passed around at that higher energy state, and then it is collected and captured, if you will, and harvested in a way that can do work. So here's another representation of the excited electron bouncing around our little pigment molecules until the electron is then transferred to an electron acceptor. So chlorophyll, as this electron is bouncing around, it's losing an electron, which means chlorophyll has become oxidized because it has lost an electron. So in our two processes, we're going to come back to this whole idea of excited electrons, but let's just go back and make sure we understand the basics of the two stages of photosynthesis. So we have the light reactions, which obviously require photons to strike them into, we're going to capture the energy from those excited electrons. And then we have the light independent reactions. When I was in college, we called them the dark reactions, or you can call it the Calvin cycle. And yes, this is because the scientist who discovered or elucidated what was happening in this reaction, his last name was Calvin. So you can see that these two reactions are linked because the light reactions produce ATP, but the Calvin cycle uses ATP. And the light reactions produce electron carriers, NADPH, and the Calvin cycle uses the NADPH. Now, remember that the light reactions need water. That's not listed here. While the Calvin cycle uses carbon dioxide. And this is the step where we actually make the glucose. Let me make a little note here. H2O is needed. Okay, so here's a diagram representing the two things, the two sides, light reactions and the dark reactions. And as you can see, I have on here what we had represented in words previously, that water is used in the light reactions, carbon dioxide is used in the Calvin cycle, oxygen is produced in the light reactions, and our sugar is being made in the dark reactions. Okay, now let's get into some specifics here. Light reactions are occurring in the grana of the thylakoid membrane. And as we've already said, we're splitting water, we're releasing oxygen, we're making ATP, and we are reducing an electron carrier to make NADPH. Now, if you remember back, we have had We have had cyclic phosphorylation, and we have also had oxidative phosphorylation. Here we have a non-cyclic photophosphorylation. But do remember, anytime you see phosphorylation, we are using fancy words to say that we are making ATP. So in the light reactions, this is a non-cyclic photophosphorylation, making ATP. So we're using photosystem 1, as you can see here, which has specifically chlorophyll P700 as its reaction center. We also use a second photosystem that is not within the photophosphorylation. It uses chlorophyll 680 as in its reaction center. So, what is happening here? So let's start at the beginning, and we will walk step by step through what is happening in photosystem 2 and 1 with our non-cyclic photophosphorylation. So, as we've already said, the light comes in, a photon strikes our pigment. We excite an electron. The electron is passed from pigment to pigment until it's finally passed to P680 in photosystem 2. Now, as that happens, because that electron is excited, it heads up to this primary acceptor. But P680 needs to not be oxidized, or it will not be able to do the step again. So water comes in and is broken down into 2 electrons, 2 hydrogen ions, which you might think of as 2 protons, and the oxygen. So I'm even going to make that note here. These hydrogens, think of them as protons because we rip the electrons off of them. So the electrons reduce the P680 because this electron is now up here at the primary acceptor. And so now P680 is ready to do the process again. But in the process of that, we have now sent an electron up to the primary acceptor. So we have reduced the primary acceptor. We have formed oxygen. We have released some protons or some hydrogen ions. And we have broken down this water molecule in the process of doing those steps. All right? Now, photosystem 2, this primary acceptor, then sends, remember, electrons do not want to stay at their high energy state. They want to fall back down to a lower energy state. So what is happening here is we have these plastoquinones and the, I forget what PC stands for, it's another plasto compound. Plastocyanin is the second, it's the PC compound. We send it down an electron transport chain, just like we saw in cellular respiration. So we pass that electron down to plastoquinone, to the cytochrome complex, and to the plastocyanin, and in the process, the energy that that electron is giving off or losing as it falls back down to a lower energy state is collected by this group of proteins that are embedded in the thylakoid membrane to make ATP. Now, notice, we are representing photosystem 1 and 2 in a way that helps you understand where the energy is. As this electron falls back down, it is now striking P700, which is another chlorophyll molecule. But notice that it is, if we drew a line across the energy state, it is not at the original level as it was when it was passed to P680 in the first photosystem. So we see that P700 is now struck by the electron, and that electron is excited and is passed again to a primary acceptor. Now, light can also strike directly in photosystem 1 and cause this process to begin. When that happens, when P700 is directly stimulated, we do not have a production of ATP from that photon of light. It directly goes up to this primary acceptor and is passed to this molecule, FD, which is ferredoxin, and goes down another electron transport chain where we use those electrons to reduce NADP to NADPH. So out of photosystem 1 and 2, we have now made ATP. Now, let's go back to the middle part here. Remember, I mentioned to you how this was similar to cellular respiration. This is another example of chemiosmosis. As this electron is falling down, we over here have these hydrogen ions. So those hydrogen ions are going from the inner part of the thylakoid across the membrane into the outer part through an ATP synthase, which makes this ATP. Over here, as the ferredoxin is passing those electrons off, we're using that to generate NADPH, which is an electron carrier. Let's look at this in another way. So imagine that a photon of light is like a giant hammer on this seesaw. And when the photon hits the seesaw, it throws this ball, which is an electron, up to this higher energy level. Well, we can't throw the ball here. That's not a possibility. But we can put the ball down and allow it to roll down and allow this energy to be collected in the form of ATP, where the ball comes and another photon of light hits it and shoves it up to this highest energy level. Okay? We can then put that electron in an electron carrier in a DPH. So as you can see, photosystem 1 and 2 work together and are needed to work together in order to make both an ATP and to reduce an electron carrier molecule. Now, your book talks about a cyclic photophosphorylation. Remember that what we have just been talking about is non-cyclic photophosphorylation, and that is what normally happens, especially in eukaryotes that we have discussed. However, there are some bacteria and there are a few algae in the protist group that do a cyclic photophosphorylation. And in this case, we only have one photosystem being used. Photosystem 2 does not exist in those organisms. And in those cases, we still have a photon of light hitting our pigments, exciting the electron, and sending it up to the primary acceptor, and then sending some of it down. Actually, we don't use ferredoxin here. Ferredoxin is over here. And we're only generating ATP. Now, because we're only generating ATP, we do not need water. We do not have the same electron transport chain where we can generate both an electron carrier and an ATP. All we're able to generate is an ATP. We're going to see that if you only generate ATP and you do not generate an electron acceptor, you are not going to be able to complete the dark reactions or the Calvin cycle. Okay, just to hit this one more time, we're using similar words that we have used in cellular respiration: chemiosmosis and phosphorylation. In this case, we're talking about photophosphorylation, which is saying that we're using the energy of light to make ATP. So, the electron transport chain between photosystem 2 and photosystem 1, as we have seen, pump protons into the thylakoid. And so those protons diffuse through ATP synthase to make ATP. Remember, these protons that we're talking about are coming from the water that is being reduced when we need those electrons in photosystem 2. Chloroplast and mitochondria are both generating ATP using chemiosmosis, okay? Both are using the same idea of this ATP synthase. The protons that are happening or occurring from another step are able to fall down their concentration gradient from a high concentration to a low concentration, where we can make ATP using that concentration gradient. But remember, the sources of energy are very different. The source of energy when we're talking about photophosphorylation and photosystems 1 and 2 is the light energy that is exciting those electrons as they're being passed along between the photosystems. In cellular respiration, the source of the energy is the breakdown of an organic molecule, specifically glucose. Okay, so here is just comparing and contrasting the location of where chemiosmosis is happening. Both of these electron transport chains are embedded in a lipid bilayer, a membrane like we've talked about, and we have multiple proteins that are embedded in there that are passing those electrons along, but one is in the cristae of the mitochondria and the other is in the thylakoid of the chloroplast. But we have the similarity of both our electron transport chains, where we have a high concentration of hydrogen ions, and those hydrogen ions or protons are falling down their concentration gradient through the enzyme ATP synthase, which is generating ATP. Here is a better picture that shows you all of the specific embedded proteins in that membrane. So here is photosystem 2, our cytochrome complex, including plastoquinone, plastocyanin, and the cytochrome, and photosystem 1 with ferredoxin and ATP reductase. And also you can see the ATP synthase. Now notice, outside of the thylakoid space, we're sending off our electron carrier and we're sending off our ATP to participate in the Calvin cycle. Okay, we often call the Calvin cycle the dark cycle or the light-independent reaction. That is because we harvest the energy in the light reactions and we send the products of that reaction, which we now know to be ATP and NADPH, over to the Calvin cycle where we can make sugar. So this is happening in the stroma, and we need both NADPH for its reducing power and the ATP for the energy. So this does not directly require light. It can happen anytime. However, because we need the ATP and the NADPH that are made in the light reactions, the Calvin cycle is not going to operate very long in the dark because it will run out of these two substances. So we have 3 phases of the Calvin cycle, and just like we've talked about with the Krebs cycle, I am not going to ask you to draw specifically the Calvin cycle or give me the names of all the intermediate steps within the Calvin cycle. You will need to know the 3 phases. Those are carbon fixation, reduction, and then we need to regenerate the carbon dioxide acceptor, which is Rubisco or RUB. So here you can see the Calvin cycle specifically. So at the last step of the Calvin cycle makes this compound, ribulose bisphosphate, which we are going to call RUBP or Rubisco. Carbon dioxide comes in through into the leaf, in particular of a plant, and is able to combine with RUBP, which makes this little intermediate here. This is where the carbon fixation happens. So basically, carbon fixation is taking the RUBP and adding a CO2 to it. That doesn't hang around for very long, and we make 3-phosphoglycerate. Now, as you can see, we need 2 ATPs to turn this cycle one time. So the first ATP comes in, and we make 1,3-bisphosphoglycerate. We then need to reduce that molecule, and we end up with glyceraldehyde 3-phosphate or G3P. This is the reduction phase. As that phase is happening, we actually make glucose after glyceraldehyde 3-phosphate. Other organic compounds might be made, but we end up making a sugar. The products of that are passed on over. You can see here we need another ATP where we put 2 of these guys together to make an RUBP or a ribulose bisphosphate that can fix another carbon dioxide molecule that will come in and turn the cycle again. So notice, if we do not have ATP, and in this case 2 ATP, or 6 plus 3, 9 total ADP, and 6 electron acceptors that have been reduced, we cannot turn the Calvin cycle to make 1 molecule of sugar. So, let's just review all that we've talked about in photosynthesis. We have 2 steps, the light reactions and the Calvin cycle. The light reaction happens within the photosystems where we think of chlorophyll being, and it requires water. The photon of light comes in, it strikes the photosystem, and we need the water to actually replace those electrons that are being excited off of the chlorophyll, which leaves us with protons. We then are able to, in the process of passing that electron along and exciting it a second time with another photon of light, create an electron carrier, NADPH, and also to create an ATP. So we're capturing the energy from this photon of light. Those two substances head over and are then also combined with carbon dioxide, which comes into the leaf, which combines to the RUBP, and we start turning the Calvin cycle, which requires both ATP and NADPH to complete the cycle. So we're taking the carbon dioxide and we are ending up making sucrose, but we can also use the sucrose to build starches, amino acids, fatty acids, and many of the different substances from biosynthesis. Because cells are great recyclers, we send the ADP and the NADP back to the light reactions to pick up either a new electron or to be put back together in the case of ATP and inorganic phosphate from another turn or more photons hitting over here in these photosystems. So, remember too that we have chemiosmosis happening in the light reactions that are very similar to cellular respiration. In Calvin cycle, we are building a molecule rather than tearing it apart, so it requires energy. If you have any questions about photosynthesis or cellular respiration, as always, you can ask me in class. You can send me an email. In this case, you can also ask a question within the discussion board that you will see below this link within Canvas. I hope you have a great day, and I look forward to seeing you soon.